Hi, everybody, it's Miss Bear, and today we are going to talk about macromolecules. There are 4 different types of macromolecules. You guys have seen this in the other videos that you've watched, and hopefully you've recognized this in the reading that you were supposed to be doing. But there are 4 different types of macromolecules that we're going to talk about today. And these are going to be broken up into 4 different videos, starting with carbohydrates and lipids and nucleic acids and amino acids. I definitely encourage you to have the following structure in your notebook. For each macromolecule, so that's going to be 4 pages in your notebook, I recommend that you use a page and have the following characteristics of your macromolecule on that page. So you're going to have the primary characteristics, so the things that this molecule can be recognized by or their behavior in biological systems, what these molecules are made of in terms of their atomic structure, and then what monomers are these structures going to be made of? What monomers are these structures going to be made of? So monomers are going to be monomers. Monomers are going to be parts or pieces of larger macromolecules, right? Parts or pieces of larger macromolecules. So it's going to be really important. I'm going to use the term monomer and then a polymer. Polymer is going to be a large molecule made of smaller pieces. So it's going to be really important that you guys know these terms while I'm talking about this lesson today. So again, I encourage you to have the structure for every macromolecule that we have. So for carbohydrates, the macromolecule name is going to be carbohydrates. That's going to be your first page. You're going to have the primary characteristics of carbohydrates. Underneath that, the atomic structures, the monomer structures, and then the variations or the types of macromolecule that you can have from this particular brand of macromolecule, right? So let's get started. Today we're going to start with carbohydrates. Now, carbohydrates are primarily used for quick energy, and they are building blocks of other molecules. So just a quick reminder, when you see a structure like this that doesn't have all of the character or all of the atoms drawn in, each line or each bend in this structure is going to represent a carbon. I know it can be kind of hard to visualize that sometimes, but just remember that each bend in a structure is going to represent a carbon. And this is our glucose molecule. Glucose is going to be all over the place. You guys are going to get so tired of talking about glucose in AP Bio. And if you've taken my class before, you know that glucose is very, very important to biological systems. And one of the reasons glucose is important to biological systems is because of these hydroxyl or alcohol groups. Okay. Now, what do these alcohol groups do? Well, if you remember, oxygen is highly electronegative. So when you put glucose into a solution, remember solutions are primarily water. So for example, when you eat our chocolate chip cookie, right? Chocolate chip cookie that we were talking about at the beginning of this unit. That chocolate chip cookie is going to be digested into its macromolecule components. So those macromolecule components that we talked about are carbohydrates, lipids, fats. Lipids and fats are the same thing. So carbohydrates, lipids, nucleic acids, and amino acids, which are proteins. This glucose is going to be the first thing that ends up being digested. It's actually going to be sucrose that is digested, but we're going to talk about that in a second. But one of the ways that we can get energy from this molecule is by breaking off these oxygens. So if you guys remember, based on electronegativity, this structure is going to be highly electronegative. It's going to have a lot of energy bound up in it. All of these bonds, these carbon-oxygen bonds, are going to have a lot of energy attached to it. You're going to read in chapter 5 and in the next parts, the next unit, about energy and how we actually quantify energy, but you don't need to worry about that right now. All you need to worry about is that these molecules, these sugar molecules, have a lot of energy. So if you remember when we were talking about water and polarity, this hydrogen is going to be equally attracted to both of these oxygens. So this is going to pull off the hydrogen and you're going to get a hydronium ion. And then that's going to leave this oxygen with a negative charge. That negative charge is going to allow it to create more chemical reactions, and it's going to be a very reactive molecule overall, right? So this is going to be true for all carbohydrate monomers. We call these carbohydrate monomers monosaccharides. And these carbohydrates are primarily going to be made of carbon, hydrogen, and oxygen. Now, the interesting thing is these carbon structures, these rings that you're seeing over here, they create a very stable backbone. So you're going to have a very stable carbon backbone, usually made of about 6 carbons, very stable carbon backbone. But then all of the oxygens on the outside of this backbone are going to be very reactive. And so the backbone itself will stay in place usually. It'll get broken up sometimes into 3 carbon chains. And obviously I'm not drawing all of the hydrogens here, but these carbon backbones are going to be very stable. They are not going to be as reactive. The part that is going to be reactive is these oxygen or these hydroxyl groups on the outside. Okay. You'll see here, these are 3 different types of carbohydrate monomers. We have glucose, which is going to be used in a bunch of biological systems that we're going to talk about in the next unit. We have galactose. Galactose is a different type of carbohydrate sugar. Then we have fructose. Fructose is going to be that primary sugar found in fruit. Okay. There are 3 types or variations that you can have of these monosaccharides or of these carbohydrates. You can have monosaccharides, which are these guys over here. Monosaccharides are a single unit of a sugar. So glucose, fructose, these are going to be monosaccharides. A disaccharide, so this is actually an example of sucrose, or an example of a disaccharide is going to be sucrose. Sucrose is your table sugar, right? Sucrose is a molecule of glucose and fructose stuck together. This is really important. This is really important because our bodies can't necessarily digest these huge macromolecules. Our bodies don't have the ability to extract energy from large molecules like sucrose or even additionally polysaccharides like starch, right? Polysaccharides are going to be big old long chains. Each one of these little chains here is going to represent a glucose molecule. Each one of these individual links is going to be a glucose molecule. So when you eat something that is really starchy, for example, bread or corn, things that are really starchy, it's going to be really hard for your body to digest that. And we have to have separate enzymes. We have to even sometimes get bacteria involved in order to digest these starches because our bodies can only digest monomers. We can only get energy from glucose. So it's going to be really important that you guys can kind of recognize, okay, when am I seeing glucose? When am I seeing another molecule? But you're not going to have to memorize these structures. It's just important that you understand their characteristics, right? And understand the difference between a monomer, a dimer, and a polymer, which in this case is a monosaccharide. Saccharide means sweet. This term here, saccharide, when something is saccharin, it's almost overly sweet. So a monosaccharide is a single sugar, a disaccharide is 2 sugars, and a polysaccharide is multiple sugars, right? Multiple sugars. And the difference between glucose and fructose, if you guys were curious about that and you're like, okay, they look pretty similar, right? They look pretty similar. One of the big differences between glucose and fructose is that it is a pentose sugar versus a hexose sugar. So this fructose structure is going to have a pentagon structure where glucose has a hexagon structure. And as we've learned in this class, changing the chemical structure of something is going to drastically change its chemical characteristics. So this is the difference. This is what keeps us from being able to digest fructose in our food. We can only, and when I say digest, I mean extract energy from. We can only extract energy from glucose and not from fructose. Here's another example of a polysaccharide. So the first polysaccharide that we saw was starch on the last slide. This is another example of a polysaccharide. And this is the molecule glycogen. In the middle of this molecule, we have a protein, and this protein is essentially going to hold on to hundreds of glucose molecules, hundreds of glucose molecules. If you zoom in on this right here, what you're going to see is a bunch of hexagon structures. A lot of times I'll just draw glucose as a hexagon, and that's indicating that it's that sugar molecule. These are hundreds of glucose molecules stuck together. And this is a molecule that is going to be used primarily for energy storage in our liver. So when you eat a bunch of glucose, a lot of that glucose is going to be stored as fat, but some of it is going to be stored as glycogen in your liver. We're going to talk about this more when we talk about digestion and we talk about how we use glucose. Here's the thing. In order to make a dye or a polysaccharide, we have to perform a chemical reaction. Now, this is not part of the carbohydrates. You can put this at the end after your 4 pages of notes. This is going to be true for all monomers and all creating polymers of all kinds, right? It's true for sugars, so it's true for making a polysaccharide out of monosaccharides, but it's also true when we're talking about fats. It's true when we're talking about nucleic acids. It's true when we're talking about amino acids. So this is a very specific chemical reaction where you have a molecule on this side, on this left-hand side. You've got carbon attached to oxygen attached to hydrogen. And then you've got another molecule on the right-hand side. You've got another molecule on the right-hand side with 4 carbons, an oxygen, and all these hydrogens, right? If you guys hopefully notice, this is going to be your hydroxide group, and this is going to be your carboxyl group, your carboxylic acid. So already you know that this structure is going to have acidic characteristics, and this structure is going to have polar characteristics. You might also notice that there is a methyl group over here, not necessarily relevant to our discussion, but it is important to be able to recognize, right? So this is going to be a non-polar region because of those carbons and hydrogens locked in together. Remember, that creates non-polar or molecules that don't have a high amount of difference in electronegativity. All right, that's a little bit of a review for you guys. The chemical reaction that we are concerned with, though, is this guy right here. So the molecules highlighted in red are highlighted for a reason, because they're going to do this really interesting thing. I've said before, oxygen is highly reactive. So when you put it into water, or when you put it into a reaction, when you put it into close proximity, or maybe you heat it up in order to catalyze a chemical reaction or to get a reaction going, it's going to do this really interesting thing. So what we have seen here is that these molecules, you have 2 hydrogens and an oxygen, have broken off of their original structure to form water. But what that leaves you with is, and I'm just going to draw part of this chemical structure, I'm not going to draw the whole thing. That leaves you with oxygen double bonded to carbon, and then an oxygen with a negative charge. This oxygen is going to desperately want to create a chemical reaction. Oh, I misspoke. That is not oxygen, because the oxygen broke off and now is part of the water there. So you're not going to have an oxygen bonded there. In fact, you're going to have a carbon that is now missing a bond, right? You're going to have a carbon that is missing a bond. It's going to desperately try to fulfill its octet rule. right? This has to do with electronegativity. This has to do with the fact that this hydrogen is just going to be as attracted to this oxygen, and that can actually pull it off of the original molecule and make it a water molecule. But now we've got this carbon that is missing a bond. And we've also got this oxygen on this side, and this is what I was drawing before. We've got an oxygen on this side that is also going to be missing a bond, right? If this is the rest of the molecule, this is the rest of the molecule. You've got an oxygen that is desperately going to try to fulfill its octet rule, right? It's going to be wanting to fulfill its octet rule. Come on. There we go. How's it going to do that? It is going to create a bond with the molecule next to it. It's going to create a bond with a molecule next to it. And that's what you're seeing here. This big old long red line that I just drew is this single bond right here, where we have now bonded oxygen to the carbon and created a larger chemical structure plus water. Again, there's a lot of deep chemistry. You can do extra research to understand the nitty-gritty chemistry of this, but this has to do with the electronegativity of these atoms and their desire to fulfill the octet rule, right? The electronegativity is going to pull oxygen and the two hydrogens off and create water. It's also going to create open octets for carbon and oxygen, and they're going to try to fulfill those octet rules with whatever molecule is most readily accessible, right? So in this example, we're taking these two molecules, maybe we're heating them up, maybe we're shaking them together, and this chemical process of either heating or shaking them up is going to produce new molecules. Now, if you guys remember from your videos, this type of reaction has a name. It's a condensation or a synthesis reaction. I like to think about this like condensation when you see water on a wall or when you see water on a window frame. Water is going to be created in this reaction. So when water coagulates or water bunches up into little droplets, I like to imagine that we're creating water droplets, right? We're creating technically molecules of water. And so this is a synthesis of H2O, synthesis reaction. We're synthesizing also a new macromolecule. We're combining monomers into polymers by the removal of water in these structures. Okay, synthesizing monomers into polymers by the removal of water. You can also do the opposite reaction. And this is called a hydrolysis reaction. Hydrolysis, hydro means water. Lysis means to split, right? We are splitting water in this case. We are splitting water, and we're also splitting the molecule. We're splitting the larger macromolecule. Here we have a disaccharide. I think I spelled disaccharide wrong. That's cool. Here we have a disaccharide. This disaccharide is going to be put into a solution with water. Disaccharide is going to be put into a solution with water. The water is going to create a chemical reaction. Ooh, that's not good. Okay. Water is going to create a chemical reaction with this oxygen here, right? Chemical reaction with this oxygen here, where the hydrogen is going to be attached to that, or attached or attracted to that oxygen. That oxygen is going to break off and stay attached to the original sugar, right? This oxygen is going to break this bond and stay attached to the original sugar. The water molecule is then going to take the place of that bond. The water molecule, or the oxygen of that water molecule, is going to take the place of that original bond. So you're going to have this oxygen right there. And then this hydrogen is going to take the place of the bond that oxygen just lost. And this is called a hydrolysis reaction. This is a hydrolysis reaction where you put or you mix a chemical with water. Water, again, you guys remember water is an amazing solvent. Water is going to wiggle its way into that oxygen. It's going to chemically react with that oxygen, split these two monosaccharide sugars apart, and then you no longer have water plus a disaccharide, you have two monosaccharides with newly attached atoms. All right. And that completes our carbohydrates section of the lesson. Keep an eye out for the lipid section.